MLT Case Study Series 17. A case study on the topic: Unraveling the Culprits of Foodborne Illness, a Dual Clostridium Outbreak Investigation. From Symptoms to Solutions, the pivotal role of medical lab technologists in detecting and managing food poisoning by Clostridium species. Introduction. Food poisoning caused by Clostridium species, particularly Clostridium perfringens and Clostridium botulinum, represents a significant public health concern. These bacteria are capable of producing toxins that lead to severe gastrointestinal illnesses and, in the case of C. botulinum, potentially fatal neurotoxicity. Medical lab technologists play a crucial role in the detection and identification of these pathogens to facilitate timely treatment and outbreak management. Clostridium overview. Microbiological characteristics. Clostridium perfringens, a gram-positive, rod-shaped, anaerobic bacterium, known for producing a toxin that causes gastrointestinal symptoms. Clostridium botulinum produces a potent neurotoxin leading to botulism, characterized by muscle paralysis. It is anaerobic, gram-positive, and spore-forming. Pathogenesis and clinical presentation. C. Perfringens food poisoning, characterized by intense abdominal cramps and diarrhea starting 8 to 24 hours after ingestion of contaminated food. Usually resolves within 24 hours. Botulism, C. botulinum infection, symptoms include blurred vision, dry mouth, difficulty swallowing, and progressive muscle weakness. It requires immediate medical attention. Detection and identification in the laboratory. Sample collection. Suspected food samples and patient fecal specimens are collected. Proper aseptic techniques are critical to avoid contamination. Laboratory techniques. Culture methods. Selective media, TSC agar for C. perfringens, and C. botulinum isolation requires specialized media under anaerobic conditions. Incubation, samples incubated at optimal temperatures for each organism to promote growth. Toxin detection. ELISA, enzyme-linked immunosorbent assay, for detecting toxins in food samples or patient serum. Mouse bioassay for botulinum toxin is considered the gold standard but is ethically debated. Molecular methods. PCR, polymerase chain reaction, highly sensitive and specific for identifying Clostridium species and toxin genes. Interpretation of results. Positive culture and detection of toxins indicate Clostridium contamination. Molecular typing can further distinguish strains involved in outbreaks. Case study. Background. A local hospital reports several cases of acute gastrointestinal illness following a community picnic. Symptoms include diarrhea and abdominal cramps. 2 patients exhibit blurred vision and muscle weakness. A joint investigation by healthcare and public health departments is initiated, with medical lab technologists tasked with identifying the causative agent. Laboratory investigation. Sample collection, leftover picnic foods, including a meat dish and canned vegetables, are collected alongside fecal samples from affected individuals. Laboratory analysis. Culture methods reveal the presence of C. perfringens in the meat dish and C. botulinum in the canned vegetables. ELISA confirms the presence of C. perfringens enterotoxin in fecal samples and botulinum toxin in serum samples from the 2 severely affected patients. PCR assays corroborate these findings, identifying toxin genes specific to each pathogen. Interpretation. The simultaneous occurrence of C. perfringens and C. botulinum in food samples consumed at the picnic, along with clinical symptoms and laboratory confirmation of toxins in patients, conclusively identifies these pathogens as the outbreak's cause. Conclusion and public health implications. Immediate public health interventions include hospital treatment for affected individuals, especially those showing signs of botulism, and a public advisory against consuming any food items from the picnic. Long-term, this case underscores the importance of food safety practices and the critical role of laboratory diagnostics in outbreak investigation and management. Thank you. Please subscribe for more videos.